Welcome to a video explaining excitation-contraction coupling, or more commonly known as how to move a muscle, by Rachael Parks. Excitation-contraction coupling is how action potential, which is the change in electrical potential that occurs between the inside and outside of a nerve when stimulated so it can transmit signals, becomes a muscle movement. So the brain, which is somewhere not seen on this paper, sends a signal via motor neuron and neuron synapse to a skeletal muscle fiber. The blue skittle is representing the signal. To a muscle fiber where the action potential passes over the sacrolemma, which is represented by this purple line, passing over, and is carried to the interior of the muscle fiber by transverse tubules, which are these green things. So this is the action potential, and when it passes over the sacrolemma, the transverse tubules feel it, and the T-tubules make contact with the sarcoplasmic reticulum, which is all around. It's covering the sacrolemma, which has the transverse tubules in it, and is represented by the little black dots and lines. And in the sarcoplasmic reticulum, calcium is stored, needed for the muscle contraction. At the top, we're going to bring this back. At the top of where the sarcoplasmic reticulum and the T-tubule meet, around here, there are protein channels that allow calcium release through when action potential passes through. And when the calcium is released, this triggers the cross-bridge cycle. The cross-bridge cycle is the cycle that allows a myosin head to attach to actin and continue muscle movement, but we'll get to that later. All of this takes place in the sacromere, which is the basic unit of the muscle, and inside the sacromere there are thick myofilaments, also known as myosin, and thin filaments, known as actin. And those together are both myofibrils. When these two muscle fibers attach, the sacromere contracts and allows for movement called the sliding filament model. This is just a closer look of the two myofibrils, and this is the sacrolemma outside of them both, see, and this is the thick one is myosin, and those are the myosin heads, and this is actin. After an action potential passes the sarcoplasmic reticulum, after it passes, the potential passes the sarcoplasmic reticulum, it releases calcium, and the calcium triggers, here's our calcium by the way, it's blue, calcium, and the calcium triggers a movement of the troponin and tropomyosin, which the tropomyosin are these lines around the blue, which is the actin, and the troponin is represented by this red thing. And so when you add calcium to it, calcium, go on the troponin where you belong. The actin to become available to the myosin head, which the myosin head is going to be represented by this watermelon sour patch. The myosin head has an ATP, which is hydrolyzed into ADP. So here's our ATP, and then it's hydrolyzed to create a phosphate, an ADP. And then this allows the myosin head to grab onto the actin by cocking into the ready position. That's the ready position. When the ATP is broken, the burst of energy allows for the myosin head to push the actin towards each other by creating a contraction, or a muscle movement, also known as a power stroke. So it's going to cock on. And then when that happens, they push the actin together so that you have a contraction, which is your muscle movement. To stop this movement, you must detach the actin and myosin, which happens when the myosin head grabs another ATP and breaks it again into another phosphate and another ADP, and then it releases. This is actually what causes rigor mortis, because you need energy to let your muscles loose, and there is none left when you die, so they both stay contracted. Thank you so much for watching this video, and I hope it was helpful.